welcome to Foresight's Molecular Machines Group. Really happy to have you all on. Before we start with today's presentation, I do want to do a minute of housekeeping, which is to tell you that our workshop is coming up. And so many of you actually went to our workshop last year, the Molecular Machines System workshop, and we're going to have a follow-up again this year. This is going to happen at the September 11 and 12 in San Francisco. It's again co-chaired by Adam Marderstone, Ben Reinhardt, and I'm helping facilitate. And if you want to take a look, there's a lot of confirmed participants already on the website, and you're welcome to apply at any point now. And the big goal will be that we'll be looking at some of the 2022 workshop advancements. There's a lot of videos from the last workshop already on here. It was pretty much focused on a lot of like software and simulation tools. And we'll be looking this year to try to integrate individual approaches a little bit more towards long-term visions of molecular manufacturing and so forth. So if you're into that type of stuff, and if you're interested in the more long-term goal of molecular machines, this one could be a great one for you to attend. And the application is open now. So do feel free to apply. Cool, wonderful. That's really it in terms of housekeeping. Now I'm really, really excited to have Hendrik Dietz here today. And yeah, Hendrik, thank you so much for joining. You'll be discussing virus traps and other molecular machines of the future. So we're already going to have a little bit of a future perspective in here. And I'm really, really excited to have you. I'll be in the chat monitoring questions and sharing a little bit more info about you and your work as well, so people have a little bit of context. But yeah, with that being said, without further ado, the stage is yours. And before we move to Q&A, you have about half an hour or something to present your work. Thanks a lot.
Thank you, Alison. So hello, everybody. Pleasure to meet you all. I want to talk about virus traps, other molecular machines of the future, some of our recent work. My laboratory at Technical University of Munich in Germany. I want to start spending a few minutes about what we're interested in generally. There's a sort of longer-term vision of the lab. So our mission or our dream is to be able to build artificial molecular devices and machines which can execute user-defined tasks. So of course we dream of reaching one day a degree of sophistication that could somehow be parallel, that could somehow parallel the type of functionalities that you currently see from the natural molecular machines that we find in nature. And then of course we also always have somehow an applied interest, or things that we have discovered or built. We always have some interest to also somehow use that then in science, technology, or maybe even health. So I will show some examples of those attempts in my talk. So the primary approach that we use in the laboratory to build artificial molecular structures and eventually machines is a biomolecular self-assembly. So we try to copy basically nature's principle in building molecular structures, encoding structures and sequences, polymers, which then hopefully fall to the structures which are encoded in sequences by self-assembly. We're primarily focusing on origami type research, but lately we also have moved a little bit into the novel protein design, but I won't talk about this today. So I want to, just to make sure that everybody's on the same page, I want to briefly speak about programmable nanofabrication with origami, so that you know what that is. DNA is our construction material. Just a quick reminder, DNA double helices usually form from two DNA single strands, Watson-Crick base pairings. So if you have two DNA single strands with complementary sequences, so G likes a pair of C and T with A, and so on and so forth. These strands, when they're in salty solution, they will wind around each other and build a right-handed DNA double helix. So now how can you build more complex structures out of DNA molecules? You can think of this as a form of weaving fabrics out of DNA molecules. Here's an example, schematic of such a fabric, where you have a bunch of DNA double helices arranged in parallel, and the individual DNA double helices, which are pointing horizontally, have different lengths, so that basically with such a sheet here, for example, you can approximate a certain envelope. And if you can do that, like weaving such a fabric, you can also imagine that you can approximate three-dimensional structures by simply stacking different types of sheets with different envelopes on top of each other, so that you can approximate the three-dimensional structure. So in this picture, you will see that actually there's a whole lot of colorful DNA single strands in these DNA double helices, but there's also consistently one strand which is blue, and this is to indicate that this is a very long DNA single strand, which is basically following a certain path through this fabric. And this but peculiar DNA nanotech design principle, which has become known as the origami, and was initially invented by Paul Roderman in 2006. And I'm sure many of you know about this. The key aspect and the ability to actually build such fabrics is the ability to actually connect double helices. And the key element here is what we call a junction, an immobilized already a junction. Let's take a closer look. Basically, if you have two DNA double helices they would like to connect, basically you can achieve that by simply choosing the right sequences. So let's say we have this top blue DNA single strand has a certain sequence, which is given here upside down. There's a lower DNA single strand in blue has this certain sequence. Then basically you can program the sequence as a thought experiment of this purple DNA single strand in such a way that first it has a sequence string segment which is complementary to this part, and then this sequence segment is followed by another stretch of letters which basically is complementary to this DNA molecule. And you can do likewise with the orange molecule, and with the simple rule of sort of partial complementarity to different DNA single strands in the system, you can encode basically arbitrary structures in sets of DNA single strands. And there could be thousands of DNA single strands. Actually depends on the level of complexity you'd like to encode in your structure. And then basically you can make these DNA single strands using solid phase chemical synthesis, for example, or also using biotechnological processes. And then you mix them all together in some controlled stoichiometries, and then the target structure will self-assemble. And here's a illustration of the self-assembly process, which is not meant to be a physically accurate representation of the actual process. It's more like an illustration. So again, you have all the components here, DNA along DNA single strand, colored in blue, and then you have a whole slew of shorter DNA single strands, nucleotides, which are partially complementary to different portions on this long DNA single strand. So as these molecules diffuse around and bump into each other, the entire system tries to optimize this or minimize its free energy. And one way to do this is by maximizing the number of base pairs formed. And the only solution by design that maximizes the number of base pairs is actually the structure that we encoded. And as a result, in this example, we get a nanoscale rod formed from 10 DNA double helices bundled up in parallel. And the beauty about this fabrication approach, if you want to think about it as a fabrication approach, is that it's basically, in a way, it's atomically precise, because you can plan in advance where in the context of this entire structure certain DNA bases will show up, and each of these bases can be addressed individually with a chemical modification, for example, or you can hook up a protein there, a fluorescent dye, and so on and so forth. So in my laboratory, we've been very much interested in pushing the complexity and sophistication of the types of objects that you can build using the origami ideas. And here's an overview of several structures that we have built in my laboratory over the last couple of years. And what you're seeing here are cryo structures. So this is actually data. These are not computer models. These basically are experimentally validated structures of objects, how they actually look like in solution. Everything that's blue or yellow consists 100% of DNA single strands that with designed sequences. It's actually the reason, there's no particular reason for yellow and blue. It's more like for aesthetic reasons, but there is a particular meaning of magenta. There are two magenta objects in this illustration, actually, and these have a natural origin. For example, this spherical object here is a hepatitis B virus. Core capsid has a diameter of 35 nanometer, and then in the upper left corner, there's another object, which happens to be a protein. It's a membrane pore. It's a toxin. Has 800 kilodalton molecular weight. So the key point of this slide is just to illustrate the diversity of shapes that can be fabricated using modern-day origami fabrication methods. These objects can also be structurally validated using cryo-electron microscopy. They can also contain internal degrees of freedom, although those are locked in, or basically frozen out for the purpose of structure analysis. So I spoke a little bit about cryo-electron microscopy, and that's kind of, I think, like a key tenet in my laboratory, that everything we build, we also try to validate as precisely as possible, because we believe like precision design, precision molecular engineering also needs precision validation. So I'd like to briefly explain how we do the cryo-electron microscopy, so then you can also better appreciate the meaning of these types of structures. So the cryo-electron microscopy, which is now I think, pretty popular method in structural biology, you take a sample, basically a little droplet of your sample containing the macromolecular complex that you would like to analyze structurally, and you shock freeze it very rapidly. And if you do it quickly enough, the water actually doesn't have time to crystallize. Instead, it forms an amorphous phase, like a glassy ice, so to speak. And you can put that then now frozen sample into a transmission microscope and then take photos, basically. And this is an exemplary TM micrograph taken in a cryo microscope. The gray contrast here is from water, which has not crystallized. And then you can see there are some objects contained in the ice matrix. They're immobile there, and they're kind of frozen at random orientations. We are actually looking at the same object, multiple copies of the same object, just being frozen different orientations in this thin layer of ice. And then you can go and use machine learning algorithms, for example, to pick individual particles and sort them according to who looks most alike, and then basically try to compute whether or not these individual particle images can somehow be reconciled with a common structure. And this is an exemplary outcome of such an analysis. In the center, you can see a 3D volume that has been reconstructed from a data set using thousands of single particle micrographs. And then there's this colorful spiky sphere, which is surrounding the object in the center. And that's kind of a histogram that shows how many particles have been found in a data set that have been assigned to particular viewing angles. And ideally, this sphere is fully covered. You don't have any missing viewing angles, and then you get a pretty decent resolution. So this is actually the 3D structure that we solve for this particular object. It's basically a barrel formed from DNAW helices with a molecular weight of about 12 megadalton. This is one object. By now we have solved the structure of many different origami objects. It's another one. It's a prototype of an object which we designed to look into global twistor formations, for example. And then this has a pretty decent resolution. You can see every DNAW helix in the entire structure. There's helical detail, meaning you can discern major and minor groove. You can see all the individual connections with the helices, and you can also see the defects in the system. For example, if you look here, there's a systematic defect in the structure. Something is not quite right. At least it didn't correspond to what we wanted to have there. And also the entire structure has some kind of global twistor formation. Okay. So with this resolution, you can start really to analyze all the structural aspects of the structure in quite some detail. For example, yeah, I'm going to show a zoom in, which I find particularly nice. So here you see two DNAW helices. You can kind of start to see the bumpiness of the backbone, which we attribute to the individual phosphates in the backbone of DNAW helices. And there's a connection between these two DNAW helices, and that's actually a single covalent bond that holds together these DNA single strands, and you can see it. I think that's kind of nice. Then when you have sufficiently detailed cryo-electron microscopy maps, you can also attempt to construct atomic models of your design structures. So in this illustration, how we do that, we use, for example, energy MD, which is a method or like a computational method, a model that has been developed by Alexei Aximentiev, who also spoke here, I think, in this group some time ago, to predict the 3D structures of the origami objects. We can use that to predict an initial model. And then what we do is we take the cryo map that we determined experimentally in a initially low pass filtered version, meaning we artificially reduce the resolution, and then we fit the initial model into the electron density map. We called it shrink wrap fitting because it looks if you were shrink wrapping the electron density map around the atomic model. And then in the end, you get a basic atomic model, which is really fitted into experimental data. And then you can look at your structure in atomic details and then quantitatively analyze the details in terms of where are the individual bases. So these are two atomic models that we construct this way from two different data sets. That's the version 1 of this object that I already commented on. They had this global twistor formations. So we analyzed the structure in detail, and then we went back to the drawing board and made a version 2 where we removed the global twistor formation, which you can see here on the right hand side. Then also the structure looks much more orderly. We removed systematic folding defect, and it looks now more straight. And this is basically an illustration of the fact that you can really do engineering. You can set some specifications that you'd like to achieve with the target structure, and then driven by data, you can go back and refine the structure so that it actually meets the specifications. Okay. Now, also For a long time, we've been interested in simply pushing the size of the origami structures that we can build. So we've been always basically trying to build ever larger objects, including more and more DNA single strands, just to explore where the limit of the self-assembly modality actually is. Some years ago, we attempted to build this dodecahedron, shown on the top right as a model. It actually worked. Didn't have a particularly high real yield, but we could actually make it. And it was basically a pre-programmed finite size 3D assembly, which contains in total 50,000 DNA single strands and 220 copies of a viral genome. With this composition, it had a total molecular weight of about 1.3 gigadalton, which is quite massive. But that was an achievement at the time. But as I mentioned, the yield wasn't particularly great. So we refined these assembly methods and came up with a new approach to make larger DNA assemblies, which we call the programmable icosahedral shell campus. On this slide, all the objects you can see here, everything that you see is data. So these are all cryo-electron microscopy maps that were determined experimentally. You can see in the top row, these triangles. So these are assembled from DNA single strands. These triangles have particular geometric features. So if you look on the top left, for example, you can see that the edges of these triangles, they're beveled. There is a certain bevel angle. And there are also topographic features encoded in the edges, which are shape complementary. And with these geometric elements, we can get these triangles to self-assemble into higher order shells, which again have finite size. Like for example, if this triangle subunit self-assembles into an octahedron, and it only makes octahedron with 95% yield. This is because the geometry doesn't allow to build any other closed shell in an octahedron. If you reduce the bevel angle, you have keeping the same interaction rules, you can get that triangle variant to make an icosahedron, and that's the resulting cryo-EM structure of that icosahedron containing 20 copies of this triangle. And again, if you then start playing also with interaction rules, and again, with the bevel angles, you can get these triangles to actually make even larger shells, which works so well in assembly that again, we can actually solve cryo-electron density maps. Yeah, so then here's a negative staining TM micrograph of these kind of shells, just give a better impression of the yield and so on. So negative staining is a bit harsh into the samples. You see also some objects which are slightly deformed, but overall, these are really nicely looking particles. So you can actually start thinking about doing something with these type of shells. So one idea that we explored is to basically use these shells as a scaffold for proteins. I will say in the middle, in a minute, what could be used for. So we made a new prototype where we functionalized the outer surface of these icosahedral shells in such a way that we had 60 copies of an IgG antibody with the C fragment pointing away from these shells. So you can see here this typical Y-shaped form of these IgG antibodies with the crystallizable fragments pointing away. And this assembly works so well again that you can actually solve a cryo-EM structure. So now what can you do with this? You can think of this as a proof of concept for making a structure which looks like a virus, but is actually not a virus. If you can mount proteins on the surface of these scaffolds, you can think about making programmable vaccines. I think that's also an idea that's been pursued by other colleagues in the field. And you can also think about using these kind of functionalizations as a way to target tissues or specific cell types using protein functionalities. Now, another interesting modality with these shells is the following. So this is a model now. The way we fixed the antibodies actually was by displaying antigens and controlled distances on the edges of these triangles. So you can modify the system in such a way that you assemble these icosahedral shells at conditions at which they are stable. Then you add these antibodies so that they can populate all of these antigen sites, which are indicated here in red. And then you transfer the system into conditions where actually the icosahedron itself is not stable. For example, ionic strength conditions, like you would have them in blood. Then what happens is that the system is still held together by the antibodies as staples, but the structure is kind of spring-loaded. And then if you add the free soluble antigens to the system, they will replace the antibodies and the whole system will basically explode. And that's a way how you can make these kind of shells responsive to biomorphic stimuli, like the presence of certain target molecules, as a way of, for example, achieving some kind of programmable payload delivery. We tested this exemplarily. We used as a payload, we used hepatitis B core capsids, actually. So the components of the virus, we loaded these shells with these hepatitis B core capsids. These shells were stabilized using anti-dick oxygen IgG antibodies. Maybe you can kind of see this as some density on the circumference, these antibodies. And then you see the hepatitis B virus capsids in the core. And once you give the soluble dick oxygen, which is antigen to which these antibodies bind to the solution, these shells fall apart, releasing the cargo. The demonstration of this antigen-triggered payload release mechanism. Now, another modality that you can achieve with these types of shells is you can actually make the oligomerization process stop at programmable partial shell states. For example, forces systems that it only makes half octahedra or half icosahedra or 3/4 of an icosahedra. And again, this works so well that you can actually solve the cryo-EM structure of each of these partial shell states. Then you can actually use these shells to actually trap viruses. Here's an example. So we use a prototype. We functionalize the interior with chikungunya virus binding moieties and then basically mix these things together. And then you get a complex of these origami shells trapping in their cavity a chikungunya virus particle. And this works so well again that you can actually solve a structure. Now, this is a raw TM micrograph of such a complex formed between chikungunya viruses and engulfing the origami shells. And you can see all the viruses here. Basically, everyone is completely covered with the origami shells. So, and you can not only trap chikungunya, you can trap many different viruses using these kind of shell or partial shell designs. We trapped, this is basically some exemplary TM micrographs of different experiments that we did. Here you can see in half octahedra mixed with adeno-associated virus serotype 2. These are the spherical particles here, nicely trapped as octahedra. Polioviruses, endoviruses, noroviruses, human papillomavirus, SARS-CoV-2, chikungunya, rubella viruses, adenoviruses, and on the right-hand side, you can see even influenza viruses fully engulfed by these kind of origami shells. On the right-hand side, we use a different type of shell, which is more flexible, so they could adapt and mold around the more pleomorphic influenza virus particles. Now, what's really cool about all of these designs on the left-hand side is actually we use the same type of binder in the center of these shells to trap all of these different viruses. So you can think of these shells as some kind of broad spectrum virus trap. And then now, what turns out that actually by engulfing these viruses, you can prevent the viruses from mechanically interacting. You can prevent the viruses mechanically from interacting with cells. You can prevent them from infecting cells in the first place. And that's might be a way to actually make an antiviral. So we experimentally tested this idea, and I'm not going to show all of the data, but we did in vitro proof of concepts in petri dishes with live viruses, adenoviruses, hepatitis B viruses, influenza, chikungunya, adeno-associated viruses, and SARS-CoV-2 variants. And we could show that with the virus trap, these half shells, we could actually very potently prevent the proliferation of viruses. I'm going to show one exemplary data set. That's here's a dose response curve taken from an in vitro virus neutralization experiment with human cell culture. On the y-axis, we have plotted the degree of inhibition relative to a sample where the virus was allowed to proliferate unhinderedly. And on the x-axis, you have the concentration of the antiviral that was tested here. So if you only titrate in the naked virus traps without any virus trapping moiety, nothing happens, virus proliferates unhinderedly. And that's expected because without a sort of specific virus binding rule, you don't get interaction between the virus. So why would it prevent virus proliferation? Then as a virus trapping moiety, we took an antibody here that resulted in the red data. You can see at certain concentration, this antibody actually suppressed the proliferation of the virus. And if you then use these antibodies sparsely in the context of these shells, you get a system which is actually much more potent in neutralizing the virus. So this is the white data and the gray data here. And this whole data is plotted as if in terms of effective concentration of antibodies. So what happens is when you put these antibodies in the context of these shells, per antibody, you get a much more potent neutralization, almost a 700-fold, I think, in this case. So, and this comes from the fact that, of course, the shell material contributes to blocking the viruses from infecting cells. And you also get avidity effects by having multiple antibodies working together and binding to a variant. So that together gives a very strong virus neutralizing effect.
How does the complete icosahedron engulf the virus, or are you only using the open ones?
We only use the open ones. Yeah, for this, so you need to have the ability, of course, to somehow have the particle enter the icosahedron. You could technically also use fragments of icosahedron and dynamically tie them around the viruses, but we found that the more efficient when we just use half prefabricated shells like here. So then the vision is really to turn the system in some kind of plug and play antiviral. So because you can actually literally take any virus binder, it could be polymer, aptamer, antibody, peptide, host receptor domain, anything. And it really only needs to bind the virus. It doesn't need to have any neutralizing capacity by itself, but of course it doesn't hurt if it already has one. You use these molecules then to coat the interior of these shells, and then you will have a functional antiviral. And it has three particular advantages. So what I showed you is you can enhance the efficacy of a molecule which is already virus neutralizing. You can also make it more resilient against mutations of the virus, because you're no longer dependent so much on the individual binding strength of individual binders to the virus. And you can make a novel antivirus with components which have no neutralizing capacity individually. I think that's kind of an interesting idea. So we like it so much that we are embarked on a preclinical development program to actually make this a real therapeutics in the future. And this is being pursued in a spin-off that we created, capsitech.com. Okay, so with this, I'd like to move to a different part of my talk, which is about motors. So I've long been inspired by the adipose synthase. It is a movie, also quite old, actually, taken from the inner life of a cell. I think it's also 20 years old now. And you can see two copies of an adipose synthase here sitting in the membrane of mitochondria. And I guess as most of all cells on Earth need ADP, so all cells on Earth also have adipose synthase. And the way adipose synthase works is as following: as we eat, other enzymes create a proton gradient across this membrane with a higher concentration of hydrogen ions, for example, here, as opposed to that, protons as opposed to that compartment. And this non-equilibrium situation can equilibrate by a flow of protons through channels which are provided by adipose synthase. And this flow can drive a rotary motion in this adipose synthase, which in turn is being exploited to drive an uphill synthesis of a chemical reaction, which in this case is the synthesis of ATP from ADP and phosphate molecules. So I think that's beautiful and obviously very powerful if you think about the fact that actually about 2/3 of our body weight is being synthesized every day de novo by ADP synthesis. So in my case, that's about 50 kilograms of ADP molecules every day. And so the question is, can we build synthetic macromolecular motors and how can we power them? So we've played around with many different ideas and eventually came up with one that works. So which we published last year in summer. So we call this a origami rotary ratchet motor. So I want to briefly highlight the people involved in this project. So the top row are all the students, postdocs working in my laboratory who contributed to different aspects of the project. On the bottom left, you have a long-term friend and collaborator for December with his student, collaborated with us on this project. On the right bottom, you can see Raman Galsanian, who's a theoretician, and Jonas Isense, who helped us with understanding the mechanism of this motor. So the motor design actually is not that complicated after all. So it basically consists of three different components. We have what we call the pedestal. On the pedestal, there is a little element which can pivot around an axis. Then we have what we call a triangular platform, which has obstacles on it. We mount this on this pedestal so that this docking side here protrudes for this hole in the triangle. And then we also add this long yellow lever arm onto this docking side. So then we get the structure which looks like this, where this yellow lever arm can rotate around the axis in the center of this pedestal. And then it's constrained to rotation in plane by the presence of this triangular pedestal. And then it also has these obstacles in the way, which play an important role in the functioning of this motor. Now, the entire object is negatively charged. It's also specifically modified so we can mount these objects one by one on chemically modified glass surfaces. And then what we do is we can stick two electrodes, platinum electrodes, into the solution, and we just apply an alternating current with a certain frequency. That's all we do. Now, there's a micrograph of the resulting motor assembly. So these lever arms rotary arms are actually quite long. This is to facilitate tracking of lever arm motions in the fluorescence microscope. At the very tip of these lever arms, we have a lot of fluorophores, so you can see the motion of these arms. And this is now, I want to show you a movie. I hope it can somehow, you can kind of see it in spite of maybe limited bandwidth. So on the left-hand side, you can see a field of view from a fluorescence microscope. There's a bright dot here, so that's the point speed function caused by the photon emission from the fluorescent dyes at the tip, located at the tip of our rotor arm. And on the right-hand side, basically you get the temporal trajectory of the cumulative angular displacement that we obtain by just tracking the motion of this rotor arm. And that's the video frame index here. So now we let it rotate freely, and it does initially a random walk. Basically, there's no net cumulative angular displacement. But then we turn on the electric field, and somehow the behavior changes. And actually, it's probably very hard to see for you. We get a progressive, processive rotation in one direction with some randomness superimposed. I have a second version of this video, which is slowed down so that hopefully you can see it even better. So here, if you follow the individual motions of this particle, you can see it rotates. Occasionally it does a back step, but most of the time it actually really rotates in counterclockwise direction. Yeah, so it doesn't do net motion, and we don't actually give the system any information whether to rotate. It's nothing imposed by external impulse. It's just a one-axis alternating current that's flowing through the system. Then there's a particle which does a clockwise motion. There's a different particle, initially random motions. We turn on the electric field, and then it changes behavior and starts moving processively in one direction. Again, I guess it's probably hard to see from the video that it actually does that. So again, I have a slow down version of that video. You can see this one actually, all the frame by frame, it actually does a clockwise motion. So just to explain a little bit the phenomenology, what happens here is basically if we have individual particles and we have the external electrodes, or what we find is that sort of depending on how the external electrodes are oriented relative to the motor particle, the motor either doesn't move at all processively, which happens, for example, this relative orientations, or it moves clockwise or counterclockwise, depending on what the orientation of the electrodes relative to the motor is. So importantly, it's really, you locate the electrodes in a particular direction, you turn on the electric field, and then the motor rotates clockwise. Or if you rotate the electrodes, then it starts rotating counterclockwise, but you don't actually impose any information externally that it should rotate in the first place. I think that's important to stress. So what we see here is really sustained rotation of these particles in a viscous medium, which means there's work done, but it's useless work. It's dissipated in friction with the solvent. The question is, of course, well, can these motors also do mechanical work that can be extracted? So we came up with an experiment to test this. So we thought maybe we can wind a spindle. So we made a variant of this motor where we fixed a 70 basis long DNA single strand torsion spring in such a way to the motor that if the motor rotates processively, it will wind up the spring and store tension the spring. So it's like a schematic illustration of that process. We made that variant and did the experiment again, and there's some exemplary data that we got. So it turns out the motors, yes, they can actually wind a spindle and then run in reverse on stored mechanical energy. So let me take you through this trajectory here. So again, we have cumulated angular displacement here. This is time. So initially the field is off, then we turn it on, the motor runs in one direction, we turn it off, and there is no more energy supplied to the system, and the rotor immediately turns direction and runs in reverse, meaning the mechanical energy stored in the spindle is now basically used to drive the motor in the other direction. So here, multiple exemplary trajectories of that kind of phenomenon. So the motor data sheet, basically of the motor that we made here, so from our observations, we could do up to 250 revolutions per minute. Our system was limited in this case by friction of the long lever arm with the solvent. Based on the sort of limited speed, it looks like the motor can do up to 10 piconewton nanometer torque. And so comparison, 80% base delivers 50 piconewton nanometers, so that's not that far away from it. And the power output then, based on friction that it generates, is about the 200 equivalents to 250 piconewton nanometer per second. That's not a lot. So you put this into watts, it's like 250 times 10 to the power of minus 21 watts, which is a really small number. But if you divide it by the weight of the motor, it's about 10 watts per kilogram. It's actually not that bad. Although it's an extrapolation, obviously. Okay, so that was one motor prototype. We also thought about the turbines, where we can drive rotation with ion flow, something that you also see in nature. So we made a Nanopore powered DNA turbine. It took us just 8 years. And this again, collaborative work, in this case with Case Decker Lab at TU Delft, and also ICX Cementive, who helped us explain some interesting phenomenon that we observed. Then, so what we did is we fabricated freestanding solid state membranes. These were done in Case Decker's lab with nanopores in there, about 40 nanometer diameter. And then we built this peculiar origami structure, which had a very stiff crossbar, and then a motor body, or like a turbine body with chiral blades. And then from that chiral blade motor body, we have a long loop, DNA loop, which is used to suck in these objects into these nanopores. This is how the nanoturbine looks in more detail. We solved the cryo structure of the different variants that we made. We made a right-handed nanoturbine variant with a right-handed chirality with rotor blades, and we also made a variant which had left-handed chirality. It's the opposite case because we were hoping that we can also see opposite direction of rotation experimentally, and also we made a non-chiral variant, which is not shown on this slide. So then, because we spent several years unsuccessfully with single nanopores, we turned to nanopore arrays, to fabricated membranes which had arrays of nanopores, so we could basically simply see more objects in parallel. And experimentally, this is how these nanopores look like. It's just a whole 50 nanometer diameter. And then what we do is that we have these fluorescently labeled origami particles, and they're being deposited in the system and sucked into these nanopores with electrophoretic flow. And then you get Sigl views which look like this. Some of the nanopores are not populated at all, but sort of like say 50% of them are populated with particles. And then you can look in the fluorescent microscope and see where these particles move. And there's a certain fraction of these particles that do move, which manifests itself as ring-shaped signatures when you take a video and then average out all of the videos. And then you can look at individual selected particles in detail and study their temporal behavior. And this is one exemplary particle you can see here. So initially, what we do is we have on the top compartment, we have our turbine variants, and then we flow in a salty solution in the bottom compartment. And this leads to diffusiophoretic docking of these particles into these pores. And experimentally, this manifests and basically nothing's happening initially. Then you see some fluorescent signal, and then immediately the particles start rotating progressively into one direction. So that was very nice. That was very nice to see. And basically, the driving force is really like a salt gradient in this case. So you have a high salt concentration in the bottom compartment and a low salt concentration in the other compartment, and you get direction rotation out of the system. So with the left-handed variants of our motor blades, we predominantly got a certain direction of rotation. Interesting enough, you also got some particles which would rotate in the opposite direction. With the right-handed nanoturbines, we got predominantly the opposite direction of rotation, which means this direction of rotation has, at least to some degree, is really tied to the molecular chirality that was designed. And with the non-chiral blades, we get some background rotation, but no preferred rotation, which also was to be expected. The fact that some of these particles rotate still, even though they don't have a designed chirality, this took us a while to figure out, can be attributed to the fact that as these particles be stuck to these nanopores, they also deform slightly, this crossbar, which has a chirality, which can couple to the flow. And that's not controllable by the users. It goes with either left-handed or right-handed. Okay, and there's also another fun phenomenon which you observed is basically that if you went to a higher salt concentration, basically using the same type of particle, we could also switch direction of rotation. So that gave us some headache. But Alex, Cementive, he actually figured out what the molecular reason for that is. Looking at the time, I'm just going to move on. So sum up. So we made 2 prototypes of energy converters, molecular energy converters. We can also call them motors. And I think for us, what's the interesting next step would be to think about whether we can also power rotation using some type of chemical process, or use the rotation which we can already drive using electric fields, for example, to drive an uphill chemical synthesis. And of course, it would also be nice to gate the activity somehow and tie it to some events. With this, at the end of my talk, I'm already over time. Apologize for that. I want to thank my funders over the years, strong supporters, allowing us to do all this stuff. And I want to thank my team, wonderful students who made all of this happen. Yeah, and with this, I'm at the end. Thank you for listening.
Wonderful. Thank you so much. I don't know if you would like to take a quick break or if you're fine with questions.
No, it's fine. Let's go ahead.
Yeah. Okay. Wonderful. Then we're going to start with Logan. If you want to unmute first, that would be wonderful. And if you want to read along the questions, they're also in the chat, but I think it's nice for people to ask them in person.
Hey, I great talk. I've actually been following your work for a number of years, and I always love reading your papers. They're super fascinating, and I just love the 3D fun, I don't know, building block style of your research. Lots of amazing stuff. My first question was about the antibody stabilized shells. And so I was wondering, so if you put those shells, let's say that in some future iteration you were to use those as a therapeutic, I would imagine that the slow off binding kinetics of the antibodies would, without antibodies that are specific to that antigen at high concentration in the solution, it would result in eventually the shells spontaneously disassembling unless there's some level of that antibody in the solution to rebind. Is that the case, or or is there something in there that allows it to still stay stable until the antigen, the soluble antigen, is delivered?
No, I think first of all, let me thank you for your kind words. Much appreciated. Thankful to hear that. No, I think that's a valid concern. We thought about that problem. We did some experiments to this extent. So we had some of these antibody stabilized shells actually contained a low salt experiment, may even be in a container supplementary information of that paper. And we didn't actually observe such over, 2 weeks. It was still stable. My take here is that simply the bivalent nature of the IgG, even the sort of one of the epitopes unbinds, sort of the local concentration of one is still so high that we just rapidly rebind. And overall, we get effectively a very tight, but very durable binding. However, it may be the case that, I guess on a case-by-case basis, you have to look, right? If you may have a poor antibody with a high KD, then maybe what you're describing may be an issue.
Interesting. And my second question was about the rotor. So when I was reading this paper a while back, I remember this same question occurring to me and being a bit confused about the mechanics of the obstacles, because it appeared that the obstacles were a ramp on one end and a wall on the other end, which you would think would mean that there'd be a strong preference, even in the absence of an electric field, for one direction of rotation, since the ramp, it would potentially have a lower activation energy for the rotor to spin along that side of the obstacle. And I was just wondering, is that intended, or is there some reason that you still get random motion without the electric field? I guess I just wanted some more clarification on that.
Yeah, okay. This is a very acute observation. So there is historic reasons to why they have this particular shape, and also practical reasons. So first of all, making these obstacles actually was really easy. So just excise a portion of these layers and just make them, basically tilt them at an angle. That makes them asymmetric already. But historically, what we actually wanted to do initially was, since this structure then, it basically has an asymmetric energy landscape. So in essence, if you had somewhere putting energy in the system that sort of, for example, you modulate the height of the pivot point through some energy consuming way, you would expect to get directional rotation, but that would be basically like a flashing ratchet type mechanism. However, so all the things that we tried to make that happen with that variant, it just didn't work. And then somehow we looked into where we can ratchet using this unidirectional electric field, and that worked. But then the asymmetry of these obstacles actually is not important at all for the fact that these motors rotate around the direction. They could be completely symmetric, but the symmetry is broken through the superposition of the external electric field with the sort of energy landscape locally. And there, the obstacles don't need to be asymmetric, but it doesn't hurt if they are. Let me put it this way. So I think we're currently looking at other ways to modulate also the opening angle of these obstacles. You could imagine you have an enzyme, for example, that consumes ATP and also fluctuates a little bit. Then you would have flashing energy barriers, and driven by some energy consumption, you would expect that thing then to show directional rotation. But it's a very acute observation.
Thank you.
Wonderful. I'll be reading out Micah's question in the chat because there's no mic. And so he's asking, in the comparison between free antibodies versus your construct, presumably your traps each have multiple antibodies on their surface. Is the y-axis in the chart the number of total antibodies or the number of constructions, or is it the total mass?
Yeah, so that The question refers to the dose response curve that I showed. And I think what's meant is the x-axis, which showed the concentration of the antiviral agent. And it was the number of total antibodies. So it's really like it's basically the fairest comparison you can make, right? And then in the case of having the shells with antibodies, basically what we plot there is the effective concentration of antibodies is they were free to diffuse in solution, but they are not because they are constrained in bundles of 30 in this case per shell. So what you're really getting is the neutralization power per antibody molecule. And by just putting it on the shell, you make each antibody more potent effectively. And this comes from the addition shielding effect from the shell material and the avidity effect.
Wonderful.
Okay.
I'll let him tell me if he has follow-up questions and maybe I'll hug in again. But for now, we have Tomo, if you want to unmute.
Yep. So I guess besides higher effectiveness, what are the advantages of using these half shells over direct antibody delivery? And part of it's just time to therapeutic generation, right? So if you already have the antibodies, you can probably pump it into the body, Regeneron style for initially. And can we actually do something like use the size and approximate chemical properties of the typical envelope size proteins found in a viral family so that we can make it more general? Antibodies are super specific, but let's just say design something for all COVID variants or all Ebola variants by kind of having a general motif that it can clamp onto.
Great questions. So let me say a few things about the Regeneron style therapy. For example, Regenkov was this cocktail of two antibodies which were harvested from reconvalescent patients, and they got infected with the alpha variant of SARS-CoV-2, and then they quickly went through the clinical program, and the drug was approved, Regenkov. Donald Trump got it, I think, when he was first infected COVID-19. He got a shot of Regenkov, and it worked for alpha very well. But then the virus is mutated relatively quickly, right? You got different variants of concern, Delta, Omicron came, and so on. And the individual antibodies, they took the hit with respect to their binding strength to the virus surface proteins. So whereas, for example, for the alpha variant, these antibodies that were developed and where a lot of money was invested in, they were effective at a certain dose. For Delta variant and later variants, often was the case, in order to get the same effectiveness, you would have to increase the dose like 100-fold or something like this, or even larger. You can't just pump in a liter of antibody into a patient. So basically, these drugs were dead. And there were actually, I know about 17 different commercial attempts to make therapeutic antibodies against SARS-CoV-2. Few of them made it to the market. Most of them are dead right now. And the problem is really the risk of losing efficacy in the presence of virus mutations. Now, and this is the key element is that these shells provide. They make these antibodies much more resilient against mutations because they're no longer reliant on the individual binding strength so much. You get basically super stable binding through avidity. If you have 90 antibodies sticking to the same target, basically it doesn't matter if the individual antibody only has micromolar KD. As a compound system, you get basically as strong as a covalent bond. So, and then you can mutate a little bit and mutate a little bit. If more binding affinity, then also the shell system won't work anymore. We have experimental data where we went for 5 different variants of concern with SARS-CoV-2. We took not a Regenkov antibodies, we took a German commercial antibody which was developed against the alpha variant. And we tested alpha, delta, and several Omicron variants. And for all of these variants, this antibody combined with the shell, it still gave an effective antiviral, but the antibody alone basically was dead or with delta. So that's a key advantage that the shells provide, makes it more resilient against mutations. The other thing is, when you think about antibody therapeutics, the antibodies, they need to do 2 things. They need to bind and also the right spot in order to be neutralizing. Whereas with our system, you can bind anywhere in the virus surface because the shell material actually does the neutralization, not the binder. So it makes you more flexible in basically finding a system which becomes neutralizing. I think you're asking for degradation time. Should I continue answering, or as already long speech? Okay.
We also have one more question. I just want to make sure that we also answer it because we only have a few more minutes left. We have Alex Forbe with a question.
Yeah, I had a few more, but I can wait.
Okay, wonderful. Alexis, if you want to unmute.
Cool. Yeah, so much, Hendrik, for the great talk. I guess, I'm always surprised with the comparison with ATPase in the sense that it seems to be like ATPase is like really like internally driven by like a chemical fuel. And here, I guess you have like external kind of like drive. I was wondering if you had plans to like install catalysis in your motors to have them being driven by by some fuel molecule or other kind of energy sources. And then a little quick question. I'm not sure if maybe I missed it, but in terms of origami stability and clinical samples and in blood, I'd be curious to have your opinion on that. Thank you.
Okay, these are two great questions. Maybe I go for the second one first because it's kind of ties to the shells. So the origami itself, actually, if you were to take it as it has been assembled and put it into environment and physiological environment, it would immediately explode and all the parts would have been degraded quite quickly by enzymes. So we actually have to do post-processing steps to make these things durable in the body. So we coat them with certain moieties. We also chemically cross-link them using via radiation. And so we also we need to protect them against nucleases. But this is all basically worked out. It's pretty straightforward to do. Then you have durable particles. To the first question, I'm using 80% days because I run about 80% days since 1997 and it inspires me as I find it super cool and just drives a little my interest. Of course, I think, I don't know, maybe I didn't make that quite clear, but it's more only like a fun comparison. Can we at some point in the future make something which is as performant as 80% days? So I kind of just compared mechanically what we observe with our motors relative to 80% days. But of course, we are light years away of having made something as cool as 80% days. That's for sure. And regarding using some chemical fuel, yes, that's the next point on our agenda to think about ways to use chemical fuel for rotation. I must confess that a little bit with the origami, we spent a lot of work increasing the position accuracy, invested in cryo analysis. And I think we're kind of got a little bit stuck at a certain level of accuracy. And I'm not 100% sure where we can. I think we need to go for hybrid structures, which will that much better than I am because you're a protein designer. So I think there might be something in it that basically hybrid structures, which use de novo design protein components attached to maybe some DNA scaffolds that help arrange in larger scale components. That might be a way to go to use catalysis for driving motion. Does that make sense?
Yeah, totally. Yeah. Thank you. I'd love to chat more at some point.
Yeah.
That's really great work. Thank you.
Fortunately, we're at time now, but if people are interested in contacting you further, Hendrik, because I think there's a lot more questions, maybe they can message me and I'll try to go for a double opt-in. I would hate to let you go without my final question, which I always ask everyone, which is if people are in this group are excited about your work, and clearly they are, what can they do to help specifically you, your lab, your team? What's kind of on the horizon? How can people help, basically?
Can you help? Well, if you're still in the academic trajectory on one of those, you can come join my lab as a postdoc. And we'll have both of us. And the other thing is, of course, computation methods, ideas for using chemical fuels, and just talking, chatting, and changing ideas. I think that's very valuable. Just my ad hoc answer to this question, I think.
Wonderful. Thank you so much. This was really great. I see so many comments also in the chat that just congratulate you on your work. So thanks a lot for sharing it with us. And I hope it wasn't the last time that we welcome you. Thanks a lot, Hendrik.
Yeah.
Thanks everyone for your great. I hope to see you at the next. Bye.